Well, thank you so much for being here today. Thank you so much for the invitation. I'm pretty happy to be here sharing some of my knowledge about Clostridium perfringens and Clostridium difficile. So, just a brief introduction about my professional experience and everything. I'm a vet. I got my bachelor in Brazil, in Federal University of Minas Gerais, and my PhD in the same university and also in the University of Copenhagen. My main research lines are clostridial disease, vaccines, and microbial resistance. I'm currently with 4 PhD students, and I have some peer-reviewed publications, and I'm part of some books. You can find the whole all my publications in this link here in your site or the platforms. Okay, so today we are gonna talk about Clostridium perfringens infection in neonatal piglets. We have 3 main pathogens here, but we are going to talk more about Clostridium perfringens type A and Clostridium difficile. And of course, if you have any questions about Clostridium perfringens type C, we can discuss later. So, for these 2 pathogens, we're going to cover etiology, pathogenesis and epidemiology, laboratory diagnosis, which is quite a challenge, control and prevention, and in the end I will leave you with some take-home messages. So, first, let's take a look in this image that summarizes the main enteropathogens associated with the pig production. As you can see here, Clostridium difficile is one pathogen associated with enteric disorders in piglets, mostly in neonatal piglets. Clostridium difficile, it's a gram-positive bacillus that can be present as commensal in some animals, but in most animals it's not part of the normal microbiota. And it's very interesting also to see that it can produce spores, which is a very resistant form that can be in the environment for years and years. This anaerobic bacteria can also cause diarrhea in several different species, including in humans and of course in piglets. So, let's talk about the disease in piglets then. The first thing that we have to discuss to understand is the source of spores, the source of bacteria for piglets, where this bacteria is coming from, and also why piglets between 1 to 7 days old are affected, why and how these animals are affected. So, the main source of spores for the piglets are the sows. So the adult animals, they are not affected, they don't have the disease, but they are the main source of spores for the piglets. And these animals, the piglets, they can, they receive the spores by fecal oral transmission, and they can really develop the disease, they can be affected. In addition to sows, we know some other source of spores for spores of Clostridium difficile for piglets. So rodents and even humans can be a source of spores to a farm. So even rodents or humans can introduce spores in a farm, and because of that, sows can shed some spores also because humans transmitted the spores to the sows, and then our rodents can transmit spores to piglets, and then you have the disease installed in your farm. And how this pathogen can cause disease? Clostridium difficile doesn't cause disease in a normal microbiota. So to cause disease, Clostridium difficile needs a disrupted microbiota, which means mostly use of antibiotics, an animal or human that is using antibiotics. So the use of antibiotics is the main risk factor for Clostridium difficile infection for horses, for several different wild animals, for dogs, and also for humans, which is Clostridium difficile infection is a very important disease. But we are talking about pigs here, and in piglets, the main risk factor is the fact that they don't have a formed microbiota. A neonatal piglet is forming their microbiota through time. So let's understand a bit more how this microbiota that is forming makes piglets susceptible to Clostridium difficile infection. Here you can see two bars. This bar here in the bottom is the microbiota of a 2-day-old piglet, and this other bar here is the microbiota of a sow. So different colors means different groups of bacteria, but we don't need to know which color is associated with which group of bacteria. The idea here is to see how the microbiota is evolving. So as you can see, we have the microbiota of 2-day-old piglet, and then 4-day-old piglet, 6-day-old piglet, 8-day-old piglet, and so on and on and on. In the end, you can see that the microbiota of a 14-day-old piglet is more similar to the microbiota of an adult animal, of a sow, than the microbiota of a 2-day-old piglet. So each day, actually each hour after birth, day by day, the microbiota will gain more diversity, and that's the key to prevent Clostridium difficile infection in pigs. So piglets are naturally susceptible to Clostridium difficile infection, to Clostridium difficile colonization. But you can imagine then, if you give antibiotics in the first days of life, mostly in the first day of life or until 48 hours after birth, this evolving of the microbiota, the speed that this microbiota is gaining diversity will be slower than in a normal piglet without antibiotics. That's why piglets receiving antibiotics in the first day of life mostly, they are more susceptible to Clostridium difficile infection, or they are more susceptible for more time to Clostridium difficile infection than normal piglets. So let's zoom in and understand how the disease affects piglets. What happens after a piglet gets in contact with Clostridium difficile spores. So the bacteria will reach the intestine of this animal and will produce A and B toxins, which are the main virulence factors of Clostridium difficile. These toxins will act in the enterocytes, in the intestine, and in addition to the direct lesion caused by these toxins will also reach the circulatory system. These toxins will induce a huge inflammatory response. As you can see here, several cells migrating to the intestine, trying to fight this infection. This is the histopathology of a healthy, normal piglet. So you can see the normal mucosa here. And here is the histopathology of a piglet with Clostridium difficile infection. In the arrow here, you can see a lot of inflammatory cells reaching the lumen of this animal because of the effect that AEB toxin has in the intestine. In the end, these lesions and this inflammatory response will reduce the ability of the intestine to absorb nutrients. And this can lead to diarrhea. But one important thing is that some animals, actually most affected piglets, they will have these lesions here, but they won't develop diarrhea. So they are not absorbing the nutrients like they are supposed to, but they don't develop diarrhea. So in the end, Clostridium difficile infection can cause diarrhea in piglets, but it's also a subclinical disease. And now we're gonna understand a bit more why this infection is a subclinical disease, but also how important, how frequent this diarrhea caused by Clostridium difficile is common all over the world. So the first study I'm going to show you was conducted in 2013 in Brazil. We were trying to understand the main pathogens associated with diarrhea in neonatal piglets in Brazil. And for that, we visited 15 farms and we collected 4 piglets in each farm, 2 piglets with diarrhea and 2 piglets that were apparently healthy. We tested these animals for several pathogens, but I want to show you the results specifically for Clostridium difficile here. 23% of the animals with diarrhea were positive for Clostridium difficile infection or for toxins. And 40% of the herds were positive. They had at least one animal with diarrhea caused by Clostridium difficile. And another very interesting thing is that among animals that were apparently healthy, without diarrhea. 10% of them were also positive for A and B toxins. So the question we had at that moment was, maybe these toxins are just present in the intestine without causing any kind of lesions or disease. But we performed the necropsy of all these animals, all piglets included in this study. And we saw that these animals that were apparently healthy, but they had A and B toxins, they had also the typical lesion associated with Clostridium difficile infection. So these animals, they were not only positive for the toxins, they were sick, they had Clostridium difficile infection indeed, but they didn't have diarrhea. These results were further confirmed by a study in Canada also, in the same year. So John Prescott group from Canada were also testing piglets with diarrhea, trying to understand which pathogens were more or less important in Canada. And they showed that Clostridium difficile toxins were present in almost 20% of the piglets. Sometimes alone, only Clostridium difficile infection, but sometimes combined in co-infection with other enteropathogens, including enterotoxigenic E. coli or rotavirus. And they also tested some apparently healthy piglets. And as you can see here, the mesocolonic edema, which is the main macroscopic and histologic alteration associated with Clostridium difficile infection, was more common in piglets without diarrhea, piglets that were apparently healthy, than in piglets with diarrhea. Again, showing that Clostridium difficile infection is a subclinical disease also. For each piglet with diarrhea, we have between 2 to 4 piglets without diarrhea, but with Clostridium difficile infection. After these two studies, several other researchers in different countries also performed more or less the same study, testing piglets, neonatal piglets for different enteropathogens, and they confirmed Clostridium difficile as a major cause of diarrhea in piglets and also a subclinical disease. So Clostridium difficile infection is a very important disease for piglets, but also it's a very important disease in humans. This is a review from Smith, where he's kind of describing the disease in humans, explaining the disease in humans. And it's very interesting because he's showing here in this image the source of spores for humans. And the main source of spores is the healthcare system. It's a hospital infection, Clostridium difficile in humans. But the second most important source of spores for humans are animals. And they selected a pig picture here to represent animals, because we know that pigs are a major source of toxigenic Clostridium difficile spores for humans. So now we know that humans can be a source of spores for pigs, and pigs can be a source of spores for humans. The idea here is that preventing the disease in piglets is not only important for the pig production, it's also a matter of public health. So we are back in this picture here, in this image here, explaining the pathogenesis of the disease. So we can discuss a bit about the laboratory diagnosis of Clostridium difficile infection. We have two main targets to diagnose Clostridium difficile infection. The first one is to detect A and B toxins. That's a very good target. And the second target we can use is to detect Clostridium difficile itself or its DNA. A third option is to perform histopathology analysis. As I showed, we have some lesions that are strongly associated with Clostridium difficile infection. But first, these two are should be considered the main option. Before taking a look in the techniques we can use to detect A/B toxins or Clostridium difficile, let's imagine that we have two options. We can sample live animals or animals during necropsy. So we can collect swab samples or we can collect feces. So it's very important to call the lab and talk to them to see if they can receive only feces or if they're okay to make the test in swab samples. So it's very important to contact the lab to understand what technique they are using and what kind of sample you should send to them. And of course, if you are performing a necropsy, you have to know where to collect, which part of the intestine you should collect. For Clostridium difficile infection, you have to collect samples from the colon, from the gross intestine. So you can target the toxins, and we have several different methods to detect A and B toxins. Most tests, they have low sensitivity and high specificity, which means a positive result, you can really be confident about a positive result. But you have to always send at least 10 samples, because the sensitivity is low, the capacity to find a sick animal is low. So the idea here to increase the confidence in a negative result is to send several samples from the same farm. And if you're trying to detect Clostridium difficile, we have several options. The first one is GDH, which is an enzyme produced by Clostridium difficile. We can detect this enzyme using a rapid test. That's quite common in humans now. And it's very interesting because you can do it in a farm, inside a farm. It takes only 15 minutes to get the results. The problem is the specificity is quite low. So I like to see this test as a screening test. You go there, you select several animals, you test them, and the positive animals you have to subject to another test. You have to test them for example, for A/B toxins to confirm that this positive result is really infection. Because sometimes you can have a positive result here that doesn't really mean infection. Another option is the classic culture methods, isolation of Clostridium difficile. In this case, you have a very high sensitivity too, like GDH, but the specificity is still medium or low, depending on the technique. So it's another option. It's a bit slow also, but it's another option that you can use to diagnose Clostridium difficile infection. And finally, we have the third option, which is the real-time PCR. It has a very high sensitivity, close to 100% also, and the specificity, similar to the isolation methods, are medium. But it's super fast. And here in this technique, you can also know the amount of Clostridium difficile present in the feces or in the intestinal content. That's also very useful to interpret the diagnosis in that animal specifically. So, in the end, you have to know the tools you're using. You have to understand which kind of tool you're using and the performance of these tools. So then you can interpret the results carefully, considering the limitation of the tool you're using. For example, here we have to understand that we are using a low sensitivity technique, so we need to sample more animals. And here we have to understand that sometimes you can have positive results, and this result doesn't really mean that the animal is infected. And it's also possible to perform necropsy. I think it's a very good idea always to perform necropsy. And you can subject this intestine, fragments of the intestine to microscopic evaluation or histopathology. We have two lesions that are strongly associated with Clostridium difficile infection in neonatal piglets, mesocolonic edema and colitis. So here we can see a piglet with diarrhea and mesocolonic edema, which is this gel present in the colon of the animal here. And this is a normal piglet. And finally, it's always important to remember that Clostridium difficile infection can be a subclinical disease. So you have to include samples from animals with diarrhea, but also animals that are apparently healthy. Because for each animal with diarrhea, you have up to 4 piglets without diarrhea but affected. So let's talk a bit about control and prevention of this disease. This is a very interesting image from a paper from Maya Rupnik showing how the disease occurs in humans. We can see here 3 main possibilities. So if you get colonized by a sample, by a strain that is not able to produce A and B toxins, a non-toxogenic strain, you won't have the disease. If you get colonized by a toxigenic sample and you don't really have antibodies, or you're unable to produce antibodies against A and B toxins, you can develop the disease. But if you produce antibodies timely against A and B toxin, you won't have the disease too. So summarizing here, we can conclude that we need to produce antibodies in our body to prevent the action of the toxins. But that's not the same way that the disease occurs in piglets. In piglets, we are talking about a neonatal animal. So first of all, if this animal gets colonized by a non-toxigenic strain, of course this piglet won't develop diarrhea. But if this animal gets colonized by a toxigenic strain, it's impossible for a piglet to produce antibodies against A and B toxins in 7 days of life. So in a normal situation, a piglet that gets colonized by a toxigenic strain will develop diarrhea. But if we give them antibodies against A and B toxin, these animals can be protected. And that was first demonstrated by Jennifer Steele in a very interesting paper where she showed that piglets with antibodies, she gave antibodies to these piglets, and then she showed that piglets with antibodies against A and B toxin, they get protected against systemic Clostridium difficile infection, and also these animals had minimized lesions in their gut. So the idea here is we can prevent the lesions and the disease in these animals by giving them antibiotics, and one way to do that is vaccinating sows. And then recently, this paper was published in more or less 2 months ago. We tested the first vaccine available to prevent Clostridium difficile infection in piglets, actually the first vaccine against Clostridium difficile infection. So we vaccinated some sows, and we collected blood from these sows before each vaccination, and we also collect blood from the piglets after they took colostrum, 24 hours after birth. And this is the results we got. So before vaccination, no sows had any antibodies against A and B toxins. After the first dose, some sows already had some antibodies against A and B toxins, and after 2 doses, all sows responded to the vaccine and produced antibodies against A and B toxins. And then we tested the piglets, and we saw that the piglets, they had antibodies. The piglets that received colostrum from sows that were vaccinated, this red here, they had antibodies more or less the same amount the sows that were vaccinated, more or less in the same concentration. So the sows responded to the vaccine, and they were able to transfer the antibodies to the piglets.